Hi, my name is Marie-Emily Maeland. I'm originally from Stavanger, Norway, and recently started as a ZooCELL Doctoral candidate in the Telford lab at UCL. Throughout my studies, I've become increasingly interested in how a single fertilized egg develops into an adult animal with multiple cell types. Even more fascinating is the fact that some marine invertebrates undergo a biphasic life cycle, meaning they start out as a free-swimming larvae and later metamorphose into an adult. This pelagic larval stage is widespread across multiple taxa. However, it is unclear whether their larvae are homologous to each other. My project therefore aims to shed light on this dispute. One feature that is similar across the various larvae is the apical organ, an organ that is proposed to be used by these larvae to sense their environment. I will characterize the morphology and gene expressions of the cells within the apical organ of the larvae of a flatworm called Processoreus crozieri and compare it to that of other pelagic larvae. The flatworms, annelids, and mollusks are within the Lophotrochozoans, one of the main branches of the protozoans, while the annelids and mollusks share a morphologically similar and well-characterized larvae that is accepted to be homologous. The larvae of my flatworm, the Muller's larva, looks quite distinct, including having 8 lobes and 3 eyes. My project will therefore employ electron microscopy to get a more detailed characterization of the cellular morphology, subcellular structures, as well as the cellular interactions within the apical organ, and complement it with already published gene expression data for these cells. I will then compare my electron microscopy studies to those that have already been published for other marine larvae. With this approach, we hope to be able to determine whether the apical organ is homologous across these species. This may also indicate what the larvae of the last common ancestors these species may have looked like, as well as give us a better understanding of the evolution of larval forms. I can't wait to see what the data has to tell us.